Hello everybody, it's Mr. IB Biology here, but you can call me Nick. And today I'm here to help you conquer Section A, Paper 3 of your external assessment. And it is here you'll be tested on your experimental skills and techniques, your analysis and evaluation of practicals that are used within the IB Biology Syllabus. If you're taking high-level biology, this section alone is worth 10% of your entire external assessment. And if you're taking standard level biology, it's worth 13% of your entire external assessment. So if you don't do well here, it's like dropping a grade, just like that. But I want you to do well. So listen up, follow me, and I'll show you how to conquer this section. So there are 5 things that you need to be able to do. First, calculate the size of an image using the scale bar provided and calculate the magnification of an image. Second, outline the Davison-Danieli model and how it was proven false. Third, provide a description of the currently accepted phospholipid bilayer model. Fourth, deduce the stage of mitosis using electron micrographs. And fifth, calculating the mitotic index using the table of information provided. So there are 4 things that you need to be able to do. The first, estimate the osmolarity of objects using a graph that is given to you and suggest why there might be differences between the objects. Second, understand the terms hypotonic, hypertonic, and isotonic. Third, predict the physical changes of an object when placed in a hypotonic or a hypertonic solution. And fourth, how to control the variables in an osmolarity experiment. So there are 3 things that you need to be able to do. The first, predicting and deducing enzyme activity from given graphs. Second, knowledge of which variables need to be kept constant within the experiment. And third, how to actually prove a reaction occurred. This is not a prescribed practical, but it is tested often within Paper 3. So there are 2 things you need to be able to do. The first, a respirometer is a device that is used to measure the respiration rate of small plants or invertebrates, and it is listed in the guide at topic 2.8. And second, you need to know how to set it up, how it functions, and what chemicals are used. So there are 4 things you need to be able to do. The first is have a detailed understanding of this experiment, including the advantages of thin layer chromatography over paper chromatography. The second, why do leaves appear red or green? The third, you need to have the skill of drawing an absorption spectrum for chlorophyll and an action spectrum for photosynthesis. And fourth, you need to have a detailed understanding of the process of photosynthesis. There are 2 things that you need to be able to do. The first is to know what variables need to be controlled within a Mesocosm experiment. The second, you need to be able to evaluate the use of a Mesocosm experiment or be able to evaluate a given Mesocosm experiment in a diagram. So there are 3 things that you need to be able to do. The first is the use of a spirometer, how it works, and how to set it up. Second is the physiological reasons of why there may be a change in the breathing rate. And third, you need to be able to calculate the tidal volume and breathing rates from the graphs given. So there are 4 things you need to be able to do in this experiment. The first is this experiment appears more often than any other experiment within Paper 3, Section A. So you need to have a detailed knowledge on how to set up a potometer and how it works. The second, you need to be able to evaluate the use of a potometer and have knowledge of where any systematic errors may occur. The third, you need to be able to have knowledge of where a xylem and a phloem appears in a monocot and dicot plant. And fourth, how to manipulate the potometer to test for variables such as wind speed, humidity, and temperature. There are two things that you need to know. The first is that any unseen data that is linked to the core material can be used. The second is that any experiment or any scientist that is mentioned within the IB Biology Syllabus can be used even if it's not a prescribed practical. This section can be very difficult if you're not prepared, and it's very easy to lose precious marks. So if you want to know how to conquer Paper 3, Section A, please don't hesitate to contact me at my email address, mribbiology@gmail.com. Start receiving tailor-made lessons to your needs and begin to achieve the results that you deserve.